The overall aim of this procedure is to culture chicken embryo slices to maintain normal neuronal development, particularly of spinal motor neurons. This is accomplished by first dissecting the chicken embryo to remove the visceral organs. The second step is to prepare and slice the embryo embedded in an agarose block. Next, the embryo slice is carefully removed from the agarose. The final step is to transfer the embryo slice into a culture medium which is incubated for 24 hours at 37 degrees Celsius. Ultimately, immunofluorescence microscopy on thin sections of the embryo slice is used to show the presence and location of spinal motor neurons following the culture period.
So this method can help address key questions in the field of developmental neurobiology. For example, the mechanisms and molecules involved in spinal motor neuron migration.
Generally, people new to this method will struggle because a lot of care is needed during the dissection stages of the protocol.
Place fertilized hen eggs with the longest side horizontally in a forced draft incubator at 38 degrees Celsius until they develop to Hamburger and Hamilton stage 24. This usually requires approximately 4 days of incubation. On the second day of incubation, sterilize the eggshell by wiping with a tissue paper soaked in 70% ethanol. Carefully pierce the flat end of the eggshell using a 21 gauge needle attached to a 5 ml syringe, and remove 5 ml of albumin. This lowers the embryo away from the shell so that the embryo is not damaged when the shell is opened. Return the eggs to the incubator and incubate them for a